Thank you. So to move this along, it's now my honor to introduce Clifton Leaf. Those of you who were here 2 years ago remember his amazing luncheon keynote. He had just published, the book, The Truth in Small Doses, Why We're Losing the War on Cancer and How to Win It. Excellent read. If you haven't read it, I highly recommend it. He's also Fortune senior editor. I know he's on deadline pretty much this week, so really appreciate him being here. He's a cancer survivor, a longtime friend of the MMRF, dating back, I think, at least a decade. So thank you for being here, and he'll lead this discussion on the Learning Network.
Great. So we're going to introduce. I'm blocking everybody's name here, but so while these gentlemen are sitting down, I'm going to just say, as Ann mentioned, I'm the deputy editor at Fortune. I run the print magazine, the old print magazine that is 85 years old, and they always drummed into us, never editorialize, never editorialize. So what I'm about to tell you is not editorializing. It's based on solid, serious reporting. But I can say, we're about to have a conversation about collaboration and data sharing and the barriers there too. And I can tell you, after a decade of studying MMRF and getting to know Kathy Giusti and the organization and Paul and Ann, I can tell you this is a problem they don't have. It almost feels a little silly coming here and talking about the barriers here when this is a problem that Kathy laid down the law years ago, that if you want to play with us, you got to share everything. You got to come to the table and be prepared to participate with and play nice with your kindergarten friends in academia and industry. So that has always been inspirational to me, and so I just want to talk about the broader issue, which is a major issue everywhere else in the cancer establishment. So, oh, great, I got double whammy here. Thank you. All right. So, so we're going to talk about collaboration and the barriers there too. You and I have not met. Hi. You got that? Like my last name. Leif, what I was reading up about you and the work that you've done, which is remarkable, but also about your dad, who was mentioned earlier, who was a real pioneer in the myeloma community, basically first real treatment that was put on the map. But the other thing where he was a pioneer was, I was reading up that he put himself through medical school basically by playing poker on the Trans-Canada Railroad. And winning and taking money from porters and waiters and probably patrons. So he really was the first to really gamble out of medical education, right? Is this... Yeah,
and his father was a Lutheran minister.
Yeah.
To this day, my grandfather doesn't know it.
So he doesn't know about any of this, yeah. It was, anyway, fascinating reading. Yeah, more?
Go over here.
All right, multiple...
That's for you. Your mic is
All right.
not working.
I have multiple mycorrhoma.
I can tell you a story about my father's first patient with multiple myeloma that was treated with melphalan, and it's kind of a story like Lori's. This was a 49-year-old truck driver in Houston, Texas, and my father treated him, and he didn't know how to use the drug, and the patient had a lot of complications. But eventually, the patient went into a complete remission. And my father did a bone marrow on him a year later and couldn't find any disease. Unfortunately, he was in Texas and he got into a gunfight and he was shot and killed. My father convinced the widow to have an autopsy performed to see if he could find any evidence of disease. And he looked everywhere and there was no disease. So back in 1960, my father thought that he was this close to curing multiple myeloma with melphalan. And, of course, that didn't happen. And as I was entering the field, he told me that we needed two things. We needed to have better drugs, and we needed to have a better understanding of the biology of the disease. And I think you heard from Kathy this morning, since my dad retired, and because of the help of the MMRF, we have those things, and it's made a huge difference.
See, I thought you were going to say we need better drugs and we need gun control. But I didn't know where that was going. But anyway. Now, if we can start out, so now you study MYC. So this is an oncogene that's prevalent in myeloma, but also in lots of other cancers as well. And there's this huge community of researchers around the world who study MYC. It's actually one of the more collaborative communities, it strikes me, from what I've been able to tell. But how have you been? How has it been? What are the barriers to sort of finding this community of fellow MYC researchers, and what have you learned from them in terms of your research on myeloma?
So Harold Varmus, who was the director of the NIH then, of the NCI, got his Nobel Prize for basically studying MYC, which in a virus causes tumors in chickens. And it's been such an important oncogene since that time. And actually, in 1982, they found out that mouse myeloma was caused by MYC. And so people looked and they studied human myeloma to see if they could see the same thing, and we didn't see it. And really, it was a result of the Multiple Myeloma Genomics Initiative run by the MMRF that we found out that half of patients have got a mutation that activates MYC. It's the most common mutation in myeloma, and we didn't know about it, and we wouldn't have known about its frequency if it wasn't for the MMRF. And so that's new information. That was published last year. And so to get everybody in the world to know about it and to collaborate on it has taken a lot of work. But because of that, we also have a mouse model now based on MYC, which gives us a very good mouse model of the disease. So I think to my mind, that was the biggest thing that came out of the Genomics Initiative.
And is the collaboration fast enough, or does it come through the medical literature, so you have to wait for a paper to be published and then comment on the paper? I mean, I can
give you a great story about that. So with my mentor and the woman who's now my wife, Marta, we found the chromosome translocation 414, but 20 years ago. So 20 years ago.
Wow.
And it was clear to us that there were other translocations. It was clear to us that myeloma was different diseases. But getting the community to accept that, obviously Kathy accepted it a long time ago, but to get it generally accepted, it was only until last month when the new guidelines came out for the staging system for multiple myeloma that the 414 is now a required element that has to be determined in each patient in order to determine their stage. So the pace is really slow. And then another story, when we found that translocation, we found out that sometimes patients had activating mutations of one of the genes there, FGF receptor 3. And we used a drug in the laboratory, and we showed that it killed those cells. And so you would think, wow, this is more than 15 years ago. Why haven't, to this date, as far as I know, there's not a single patient with an activating mutation of FGF receptor 3 that has been treated with a drug that targets FGF receptor 3. And it's not because I haven't tried. I ran a clinical trial of a drug that targets FGF receptor 3, but the drug company wasn't interested in testing what turns out to be 5% of patients with multiple myeloma. They wanted to treat everybody with multiple myeloma, and among the patients we treated, none of them happened to have the mutation. And I think you also heard from Kathy that the MMRF now will be doing a trial where the right drug will be given to the right patient. And I can't wait to see a patient treated and see if it works as well in patients as it does in the dish.
Now, that's a perfect transition to you, Colin, because what you do is you help model those connections through data and through your algorithms. And how do you think you can speed up this process of getting us to the point where we can find these connections, A, and then begin to make a case for testing them, to sort of make a legitimate case that says there's enough credible evidence to say this is a real connection, not an artifact?
Sure. So I think the opportunity has been created with both the sequencing of the human genome and the development of technologies that can now probe patients and their tumors and bone marrow at a much more granular level than ever before. But it's not only that type of data. It's also now our ability to gather data from many, many patients and aggregate that data coming from electronic medical records, data from genomics, data from other sources. Number two, it's also the rise of computing. We've had decades of Moore's Law that give us very fast, cheap, cloud-based supercomputers capable of crunching data orders of magnitude faster than we could have imagined, even compared to 5 years ago and 10 years ago. And then I think the third piece of this is artificial intelligence and machine learning technologies that can now probe data in unprecedented ways. So we're not just looking for patterns in data, But we can now use the data from many patients to reverse engineer computer models that are capable of matching interventions to patient backgrounds, the process variables like the molecules that move up and down, to predict clinical outcomes and even economic outcomes. And it's really the convergence of these three developments that give rise to the opportunity to turn data streams such as the Compass data stream into now computer models that can do two things. One, can discover new pathways and new drug targets, and that's already in play and from the first phase of the effort analyzing 800-odd patients. We've already now discovered, in collaboration with MMRF and the various collaborators, new pathways, new drug targets that are being validated. Number two, because that first effort is around creating new drugs and new combos, the second is to better match the drugs that are already on the market or about to come on the market to patients. And so this is through using the computer models to discover predictive biomarkers, response biomarkers, and diagnostics so that patients don't have to go through 9 failed drug treatment pathways before getting the right one. What if we could just learn from all of the tens of thousands of patients, hundreds of thousands of patients who have tried various treatments? Some failed, some didn't. We all have different genetic backgrounds. What if we could learn from that data ahead of time and therefore predict for you and for other multiple myeloma patients the first time from the data in your tumor biopsy which treatment is going to work the best for you? And that's not pie in the sky. When we started our company 15 years ago, I was 27 years old at the time, we were considered a little crazy, a little out there, because no one was trying to do that yet. The human genome wasn't sequenced, and computers were still slow and clunky and expensive. Now, I would say this is not such a big and crazy idea. I mean, it's edgy, but it works, and it's working better and better as more and more data gets collected. As you increase the N on the number of patients, the computer models get better and more accurate, and from new data that's used to then constrain the models further. So the same excuses that may have existed 10 years ago or 5 years ago, they don't exist anymore.
See, you said something at lunch today, and this is a perfect transition. You said something at lunch that flew in the face of the wisdom of my 11-year-old daughter, which is that you have plenty of computing power. You've got, more than enough. It's cheap. The cost of sequencing has gone down. The chips have gotten much better and faster. So what are the barriers then? Because you talked also about some barriers to this.
Right. So there are still barriers, and I would say it really comes down to more data sharing. And data generation is still a bottleneck, but much less so than before, right? Like 10 years ago, if one wanted to conduct a study like the Compass study, it would have been way too expensive. The technology just didn't quite work well enough. And even the computing power to make sense of it was difficult. And I think data sharing among institutions was somewhat, it just wasn't done. And so I think some of those barriers have come down. The computing barrier is no longer there. I don't think that's a credible barrier. I think the algorithms, I think we've moved beyond that. I think there's lots and lots of interventions. There's enough drugs on the market or in clinical development such that it's starting to move more to a matching problem more than we just need simply new interventions.
So that brings up, Nicholas, we were talking about biomarkers, which is your area, or one of your areas of expertise at Janssen, or Janssen, I should say. Has everyone else mastered that, by the way? Janssen? Okay, good. But we were talking about there are some 30,000 cancer biomarkers out there. Maybe none of them are, very few of them have been validated, but they're out there in the literature, proffered, I guess. And yet, you were talking about how few there are actually that are of use or for prognostic value or diagnostic value or even being able to show us progression to disease. You kind of elucidate on that?
Yeah, well, if you look at the databases, as Colin knows, there's more than 40,000. If you look in some of the biomarker data, you'll see lists of 40,000 biomarkers. That's suggesting there's 2 biomarkers for every gene there is in the genome, and this is patently ridiculous. they're not really biomarkers, you know. So, but if you look at the other extreme of the spectrum, of things that have actually been converted to clinical utility, in oncology today, there are only 20 drugs with a diagnostic test approved by the FDA, with an in vitro diagnostic test approved by the FDA to direct therapy. You know, there are many others in development. You know, you mentioned FGFR. There are a number of FGF receptor inhibitors that are targeted against patients carrying translocations or mutations of one of the FGFR genes. There's a lot of those in development, but it's still a relatively small number. And I think one of the biggest bottlenecks that we face in trying to enroll patients in clinical trials is the overall yield. You know, and so when we look at the numbers of patients we have to screen, so if I pick FGFR as an example. To get 5 people enrolled in a trial, we may need to screen 100 because 95 of those people will not be eligible. So means this is not feasible. It's not, you know, financially or for the patient. It's not a good system where, you know, 95 out of every 100 patients will not potentially benefit from your therapy. So I think the development of these new bucket trials, as we call them, these multi-drug trials run through, you know, the NCI, the Biomarker Foundation, which has 2 advantages. I mean, the biggest advantage to the patient enrolling in those trials is you're testing multiple different molecular mechanisms. These are each independent of each other, so there's a much greater probability that a patient will have a molecular phenotype that's eligible for one of the therapies that's in the bucket trial.
So when we talk about precision medicine, I mean, fundamental to that is having a biomarker that is appropriate as representative, a surrogate of that disease.
Well, in many cases, the ones that are actually approved today, really all of the FDA-approved markers really actually measure the status of the drug target. It's a mutation or a translocation in the drug target, and they're all tyrosine kinase inhibitors. So, for example, we've spoken about KRAS. You know, I think we may have mentioned EGFR. The thing we're measuring, the biomarker, is a mutational activation of the gene that we're inhibiting with the drug. We're very, very good at combining tests and drugs for tyrosine kinase inhibitors. You go to other classes of drugs, we're not as good. We don't yet know how to predict why some patients will have a prolonged deep response to immunotherapy and other patients won't respond at all. Otherwise, similar-looking patients won't at all. We're not very good, you know, in terms of proteasome inhibitors, as you talked about earlier today. We're not very good. We can't really predict how patients will or won't respond to Velcade. We can't predict how patients will or won't respond to epigenetic modulation yet. And so the magic of tyrosine kinase inhibitors, of course, is the marker is the status of the drug target, so we know where to look and it's easy to find. But we've got to learn how to do that, because if we think about immunotherapy, you're seeing some patients who are essentially being cured. You'll see patients, you know, in the melanoma or in the lung cancer area who are 5 years post-treatment with a checkpoint inhibitor disease-free. You know, stage 4, late-stage disease patients who in any previous time would not have survived more than a matter of weeks or months, and now they're living 5 years disease-free. But that's still a minority of patients, and the goal obviously is to take that minority and understand what that is and then drive that up into a much greater group of patients. But it's a very complex thing once we start getting away into these more complex drugs and they're less clear mechanisms.
You know, we all got a treat this morning when we heard Dr. Caleb, you know, hopefully our new FDA commissioner, talking about what they were looking for. And one of the challenges is the fact that the surrogate markers often don't really connect with the endpoints, the clinical endpoints. That after when you study enough patients or enough of these cases, that the effects wash out.
But I would argue in the FDA's defense, though, that where those cases have been brought forward and you have a very, very accurate marker with a drug that is highly active, they have approved those combinations extremely quickly. Where you see issues of these approvals dragging on, it's often because the marker isn't very good and your sensitivity and specificity of the test isn't great, or perhaps your drug isn't very good. But where we've been able to bring the two things together with a really high-quality test that really says who will and won't respond, and that drug is highly active, the FDA has been very rapid in approving those types of drugs.
So, Leif, how can we take this challenge and look at it through a new lens of collaboration? we have all of these purported biomarkers. We know they aren't true, that it's impossible mathematically. How do we start to make sure that we're all working together so that we're not duplicating each other's efforts and focusing on some of the most, potentially the best targets?
Right. Well, I think the talk about the bucket trials, which are the clinical trials where you sequence and you identify all the mutations and you have access to all the potential drugs, is certainly one way. And there's a limit to how many people or places can run bucket trials. You need a certain scale to do that. And the MMRF is obviously one. I think some drug companies are running their own as well. But I think that's definitely one way we can do it.
And there seems to be more regulatory willingness to run these things, to sort of sanction them. There have been quite a few of them. Don Barry has one, in breast cancer. Is the regulatory environment? Let me ask you a tough question, Colin. I'll let you take a stab at it since you're the youngest. Is it? You have the most to lose. Is it really where it should be in terms of catching up with the science?
No, it's not.
Okay, next question.
Yeah, I don't think anyone, including the people who run the FDA, I think they've been frustrated really by the ability of regulation to catch up with the science. There's a few notable exceptions, as Nick pointed out. But I actually think that randomized controlled trials are important. They have their place. They shouldn't go away. But we need to find ways to learn much faster from the real world in parallel, right? So you take the concept of the bucket trial. It's a great sort of multi-mechanism, multi-drug type of clinical study, but take that concept into the real world, where we have 10 times the number of patients to potentially learn from, and the ability to also
probably
use
100 times, right?
Or 100 times.
It's less than 1 or 2% of adult cancer patients ever get involved in a clinical trial.
So absolutely. So imagine we're getting 100 times the data, where we're seeing, instead of being stymied by accrual times, we're seeing in the real world with patients who do need to get treated. They have no choice but to participate. So now you have all these natural experiments happening in the real world, and novel and very powerful computational techniques that can even extract causality or something akin to cause-effect relationships from that data. Is it the gold standard randomized controlled trial? No. But can you still learn a lot of powerful information about what works for whom and have that progress in parallel with clinical trials? I think that's the biggest bottleneck actually we're facing.
So that brings up a really good question, because we've talked about this all day, and Leif, maybe you can talk about this, but we've talked about the incredible heterogeneity of cancer patients, and particularly in myeloma, where everyone is almost an N of 1. I mean, whether the unique biology of each of us, and not just obviously from a genetic mutation standpoint, but from all of the elements of our makeup, make us so unique. Does it ever make sense to try a one-size-fits-all, drug on a large group of patients, because you're really aggregating lots of patients with different types of disease?
Well, sometimes it makes sense when you've got a specific mutated target and a specific gene that goes, or drug that goes after that. But a lot of the time it doesn't. And that's why the study that we heard about that Lori participated in, they used each patient as their own control. They looked at how long the disease stayed away after the first transplant and compared it to how long it stayed away after the second transplant combined with the CAR T cell. And that sort of removes a lot of the heterogeneity. And so innovative trial design like that, I think, is another way forward.
I think that's a key point. And what's I think we have to see happen is a change in the paradigm of what a clinical trial is. And this is where the COMPASS trial, I think, is so important. Because traditionally a clinical trial has not been a means of learning anything new. It was designed and statistically powered to confirm or refute a pre-existing hypothesis. And you recruit the minimum number of patients as quickly as you possibly can to ask if your treatment X is benefiting this group of patients within a certain period of time. And it's very limited, the questions are very specific. You have a primary endpoint, a secondary endpoint, but you're not learning anything new. You are just confirming or refuting an existing hypothesis.
Mm-hmm.
The COMPASS trial is the reverse of that, is the inverse of that. It's not hypothesis-driven. It is large, 1,000 patients, deeply covered, and allows you to create hypotheses that can be tested appropriately subsequently.
Mm-hmm.
And you can learn something. So we will learn enormously out of the COMPASS trial in a way that you cannot really learn out of a clinical trial. And indeed, many of the things that Colin does would be viewed by traditional clinical statisticians in the traditional pharmaceutical industry as statistical sins. It's not, and I think that's the change that's really happening, is we're moving from a clinical trial being just a device to confirm or refute a very simple idea to it being a means of generating information about the individuals in the trial and understanding why individuals respond differently to similar therapies.
So that's a novel idea. So the idea of the trial is something you can actually learn from, which I kind of like.
Including the failures.
Including the failures,
absolutely.
including what doesn't work.
Yeah, I mean, my boss always used to, when we're designing clinical trial plans, I mean, still to this day, 1 in 10 compounds that go into the clinic ever make it through to a phase 3 registration. Bill Hite, who's my old boss at Janssen, basically just said, I don't care if the trial fails, but what I do want to know is why it failed.
Right.
Because if we don't understand why a drug trial or a treatment trial fails, you haven't learned anything. And you haven't improved your chances of doing something different next time. It was really an issue of understanding. It's great if it succeeds, but if it fails, let's understand why it failed.
On that, I'd love to throw it open to the audience for questions so we can keep on track with you. Sure. Mm-hmm. Oh, yeah, we can do anything you want. What? So you want to keep...
I think a couple questions and then...
Okay.
Because I know a lot of people need to leave.
Yeah. So, I'm sorry, yes questions?
Is there anyone with a burning question?
Okay.
Carol, just for you.
Well, okay, it's not so burning, but the point that you made about the COMPASS trial really being a new paradigm and how we look at things versus the clinical trials. So this could potentially change then the way the FDA is able to look at the approval of new drugs, and how do we keep that alive? How do we make something like that happen?
Yeah. I still think it will help bring more ideas that we would then be able to bring to the FDA that would be formally statistically proved. So I think it doesn't replace those subsequent trials, but let's go back to the FGF, for example, where you get about 5% of lung cancer patients carry this particular abnormality. So in order for us to show statistically that a patient benefits from therapy, if we could enrich that trial for only lung cancer patients with FGF receptor mutations, or in the case of this audience, multiple myeloma patients with FGF mutations, we have a much stronger statistical signal of efficacy or lack of efficacy. You can imagine if 95% of your patients do not carry that molecular abnormality that drives response, you're diluting your signal now. Your ability, so you have to run an enormously large trial. It takes you huge amounts more money, much more time. So our ability to test targeted therapies quickly and efficiently and not spend a lot of time, a lot of money, and most importantly, not have a lot of patients who can't respond to a therapy on a test, is we need to find those hypotheses underlying those events and then test them prospectively very specifically.
So this was the theory behind Genentech's Herceptin trial, where it limited the group of women it was testing its drug on to people who were HER2 positive, so that it at least had a connection between the surrogate, between the marker and the disease and the.
And you can run a significantly smaller and faster clinical trial, with that idea in place than basically testing all comers.
And that was the first approval for a drug.
And that was in 1990.
Yeah,
and
I mean that's a long time ago.
That was 1998.
Right.
So here we are, 2015, 17 years later, and we've had about 1 targeted therapy approved a year, because there are right now 20 targeted therapies approved in oncology.
Wonderful.
I just add to that point, and Nikki, you were bringing this up earlier. Part of the question that I actually did get directly from FDA was, tell us what the natural history is of that particular alteration on progression of disease. So it shows evidence that demonstrates there is an impact of this alteration, and then that is the backdrop to doing exactly what you just talked about, Nick, identifying those patients with that alteration that have this disease, and then treating them preferentially with a therapy that targets that hopefully.
And then when we as a sponsor bring something of high quality like that forwards, it gets approved very quickly. Where we run into problems, and in some sense, I mean, I'm a member of the industry, so I'm not as usual pointing fingers at the FDA. But at the end of the day, though, I mean, if we have a very powerful case with a targeted therapy with high response in the targeted subsets of patients, that is being, is the regulated system perfect? No, but those are getting approved quite quickly. Where we run into trouble is where our drugs are marginal or our targeting is marginal.
And this is, I think, why the COMPASS trial has a potential impact that goes far beyond the 1,000 or so patients that are in it and the knowledge you can learn from it. Because really, as we discover that we can learn more from it, it ushers in a sweeping cultural change to allow the regulatory authorities to look at new ways of doing trials and accept more the knowledge that's gained from these different types of analysis. So that was editorializing, by the way. Right.
But the COMPASS trial is a wonderful thing.
Yeah.
It is a stupendous achievement.
Well, thank my guests. Thank you very much. Okay.